Next chapter, Chemicals of Life. Now, all living organisms are composed of similar chemical compounds called biomolecules. Biomolecules are organic compounds, meaning they contain carbon-carbon or carbon-hydrogen bonds. Methane is the smallest organic molecule. The central carbon, by sharing electrons, has formed chemical bonds with 4 hydrogen atoms. Carbon is important to life because it is a very versatile element. In addition to hydrogen, carbon can also bond with a number of atoms, including oxygen, nitrogen, phosphorus, and sulfur. Carbon can also bond with itself, forming chains, branch chains, and ring structures. It is also possible for carbon to form double bonds with itself or other elements such as oxygen shown here. The carbon chain of a biomolecule is called a skeleton or backbone. And just like an animal skeleton accounts for its shape, biomolecule skeletons account for their shapes. The reactivity or properties of biomolecules is largely defined by a functional group that attaches to their backbones. A functional group is a specific group of bonded atoms that always has the same chemical properties and always react in the same way. The functional group shown here is a hydroxyl group. When it attaches to this carbon backbone, the compound ethanol is formed. There are many types of functional groups. In biology, the common ones include hydroxyl group, carboxyl group, amino group, sulfhydryl group, and the phosphate group. In these diagrams, the R's refer to where the functional groups attach to the carbon skeleton. The presence of a functional group determines the reactivity of the biomolecule. Functional groups therefore help in determining the identity of the biomolecule. For instance, when this carboxyl group attaches to the carbon backbone, a fatty acid biomolecule is formed. When an amino group and a carboxyl group join with this carbon backbone, an amino acid biomolecule is formed. All biomolecules are divided into 4 classes: proteins, carbohydrates, nucleic acids, and lipids. In this chapter, we shall discuss carbohydrates, proteins, and lipids. Carbohydrates. Carbohydrates are biomolecules that contain carbon, hydrogen, and oxygen atoms in the ratio 1:2:1. We can represent the proportions of these atoms within carbohydrate molecules with the formula CH2O. Carbohydrates are classified into 3: monosaccharides, disaccharides, and polysaccharides. The word saccharide simply means sugar. The prefixes mono, di, and poly refer to the number of sugars in the molecule. Mono means 1, and so a monosaccharide has 1 sugar unit. Di means 2, and therefore a disaccharide is a carbohydrate that has 2 sugar units. Poly means many, and therefore a polysaccharide is a carbohydrate that has many sugar units. Monosaccharides. Monosaccharides are the building blocks or monomers of all carbohydrates. Common monosaccharides include glucose, fructose, and galactose. Glucose is by far the most abundant monosaccharide. It is the energy source for cellular respiration and ATP production. Fructose is the primary monosaccharide found in fruit and cane sugar. Galactose is the primary monosaccharide in milk. All these monosaccharides have 6 carbons and therefore have the formula C6H12O6. All monosaccharides are soluble in water, forming a sweet-tasting solution. They reduce the copper-2 sulfate in Benedict solution to copper-1 oxide, and they are therefore called reducing sugars. Monosaccharides are also crystallizable. They can be linked together to form complex carbohydrates in a process called condensation. Glucose is abundant in blood and helps to maintain blood pressure. It is what we call blood sugar. Monosaccharides are oxidized during respiration to release energy. Glucose is the respiratory substrate, and all others have to be first converted to glucose to undergo oxidation. They can be condensed into polysaccharides for storage. Disaccharides are formed when two monosaccharides join together in a condensation reaction, forming what is called a glycosidic linkage. During condensation, a water molecule is lost. Condensation is therefore a type of a dehydration reaction. Common disaccharides include maltose, which is made of two glucose molecules. Sucrose, which is also known as table sugar, is made of glucose and fructose. And lactose, also called milk sugar, which is made of glucose and galactose. So you see, in each disaccharide, glucose is present. And just like monosaccharides, disaccharides are soluble in water, forming a sweet-tasting solution. Maltose and lactose are reducing sugars because they reduce copper-2 sulfate to copper-1 oxide. Sucrose is a non-reducing sugar. It cannot reduce copper-2 sulfate unless it is first broken down to its constituent monosaccharides. All disaccharides can be broken down into their constituent monosaccharides in a process called hydrolysis. The process involves splitting the molecules in the presence of water. In cells, disaccharides are hydrolyzed by enzymes, but in labs, hydrolysis is done by heating disaccharides in acid, such as dilute hydrochloric acid. Disaccharides are hydrolyzed into monosaccharides and then oxidized to release energy in cells. Polysaccharides are formed from glucose monomers joined together to form long chains. These long chains of glucose are ideal for storage. The chains can be flat or branched. Plants store starch. This may be in the form of amylose, which is straight chain, or amylopectin, which is branched. Animals differ from plants in that they store food in the form of glycogen, which is a highly branched polysaccharide which can be broken down easily to supply energy to tissues. Glycogen is abundant in liver and muscle cells. Other polysaccharides such as cellulose, chitin, and peptidoglycan serve as structural molecules in organisms. The most abundant is cellulose. Cellulose is a straight-chain polymer of glucose, just like amylose. Most organisms, including humans, are unable to break these cellulose bonds and cannot therefore use cellulose as a source of energy. Cellulose is used to add strength to plant cell walls. Chitin is a structural polysaccharide found in animals and fungi. It makes the exoskeleton of insects and crustaceans. Its unique properties are a result of having amino groups attached to the sugar monomers. Peptidoglycans are complex polysaccharides found in cell walls of bacteria. Most antibiotic drugs target the peptidoglycan to kill the bacteria. All polysaccharides are insoluble in water and are not sweet-tasting. In summary, we often classify carbohydrates as simple sugars or complex carbohydrates. Monosaccharides and disaccharides are simple sugars. Complex carbohydrates refer to the polysaccharides. Next, we look at tests for carbohydrates, and we start with reducing sugars. Now, we indicated that all monosaccharides, maltose and lactose are reducing sugars. That means they reduce the blue copper 2 sulfate to brown copper 1 oxide. To test for reducing sugars, we used Benedict solution, which contains copper 2 sulfate. So in this experiment, we will test for glucose in a banana sample. So we have the Benedict solution, a banana extract in a test tube, and a Bunsen burner. We take a small amount of the Benedict solution and add to the test tube containing the banana extract. We then place the mixture in a water bath. The color changes from blue to green to orange and brick red, indicating the presence of glucose. The same procedure is done for all the other reducing sugars. Non-reducing sugars. Sucrose is a non-reducing sugar. It cannot directly reduce copper 2 sulfate in Benedict solution to copper 1 oxide. Instead, sucrose must first be hydrolyzed into its constituent monosaccharides. This will then reduce the copper 2 sulfate. Hydrolysis is done using hydrochloric acid. So, we take a small amount of hydrochloric acid and add a few drops to the test tube containing sucrose. Boil the mixture in a water bath for 2 minutes. This will hydrolyze sucrose into glucose and fructose. Now we add a few drops of sodium hydroxide into the test tube to dilute any excess acid. Now we perform the Benedict test. Using a dropper, we take a small quantity of Benedict reagent and add to the test tube containing our sample. We boil the mixture in a water bath for about 2 minutes, and the color changes from blue to green to orange up to brick red, indicating the presence of a reducing sugar. Next is test for starch. Starch is a non-sugar and the food stored in plants like in seeds and tubers. We use iodine to test for the presence of starch. So this is the iodine solution and some potato extract in a test tube. We take a small quantity of iodine solution and add 5 drops to the test tube containing the potato extract. The blue-black color indicates the presence of starch in the potato extract. The next biomolecule is lipids. Lipids are a group of hydrophobic biomolecules that play an important role in living organisms. While the primary role of lipids is long-term energy storage, lipids are also used for protection, insulation, and lubrication. They also act as precursors for some hormones and are a key component of the cell membrane. Lipids are quite inert, allowing them to be stored in tissues. There are 4 basic groups of lipids: triglycerides, phospholipids, steroids, and waxes. Although these groups differ in many respects, they all have one common characteristic: they are insoluble in water. Lipids are hydrophobic. Hydro means water, and phobic means fear of. They are, however, soluble in organic solvents such as chloroform, alcohol, and ether. Triglycerides include fats and oils. Fats are solid at room temperature and are used by animals for insulation, protection, and long-term energy storage. Oils are liquid at room temperature and are used by plants for long-term energy storage. Triglycerides consist of two types of molecules, glycerol and fatty acid. Each molecule of triglyceride consists of one glycerol arm and 3 fatty acid arms, and hence the prefix tri. The next category of lipids are the phospholipids. These are similar to triglyceride in that they contain glycerol and 2 fatty acids. The difference is that a phosphate group is attached instead of a third fatty acid like in triglycerides. Phospholipids are immensely important, mainly due to their properties in regard to water. The phosphate head of the molecule is hydrophilic or water-loving. This means it mixes well with water. The fatty acid tails are, however, hydrophobic or water-hating and do not mix well with water. Because of these unique properties, phospholipids tend to arrange themselves so that only the hydrophilic tails interact with the watery environment and the hydrophobic tails crowd away from the water. This structure is the major component of plasma membranes of cells. Steroids are composed of 4 fused rings of carbon to which different functional groups are attached. A common steroid is cholesterol. Cholesterol serves as a precursor for synthesis of other steroids such as testosterone, estrogen, vitamin D, and cortisone. Cholesterol is present in plasma membrane where it stabilizes the membrane. The hormones testosterone and estrogen have small differences in their functional groups but large differences in their effect on organisms. Waxes are nonpolar and repel water. They are found on the cuticle of leaves and outer surfaces of animals. Waxes produced in the ears of some animals protect the eardrum. In addition, bees construct honeycombs from wax. Now we look at tests for lipids, and we do the Sudan 3 test. In this test, we will need the Sudan 3 dye and oil in a test tube. We add a few drops of Sudan 3 reagent to the test sample in the test tube. We shake the solution to mix. Pink droplets appear, indicating the presence of lipids in this sample. The last biomolecules we look at are the proteins. Proteins, like carbohydrates and lipids, are compounds of carbon, hydrogen, and oxygen. However, unlike carbohydrates and lipids, proteins also contain nitrogen and sometimes phosphorus or sulfur, or both. Proteins are made up of long chains of amino acids. All amino acids have a similar structure with a carboxyl and amino functional groups. What makes each amino acid unique is the presence of a variable or R group. The R group gives each amino acid its chemical properties. Amino acids are linked together by peptide bonds, forming long polypeptide chains with the loss of a water molecule. As these chains form, the polypeptide develops multiple layers of structure which contribute to its overall shape. These levels are called the primary, secondary, tertiary, and quaternary structures. The primary structure is the order of amino acids in the polypeptide chain. Amino acids can be likened to the letters of the alphabet. Just like thousands of words can be constructed by rearranging the 26 letters in our alphabet, So too can different proteins be created by varying the number and sequence of just 20 amino acids. Most proteins dissolve in water to form colloidal suspensions rather than true solutions. Proteins are sensitive to temperature and pH. Temperature above 40 degrees Celsius denatures or destroys the three-dimensional configuration, rendering the proteins ineffective. Acids, bases, and organic compounds also cause denaturation. Proteins are amphoteric, meaning they have both basic and acidic properties and can therefore react with both acids and bases. This property allows proteins to combine with other non-protein molecules to form conjugates. For instance, they combine with lipids to form lipoproteins. They combine with carbohydrates to form glycoproteins. They can even combine with inorganic substances like metal ions. For instance, the protein hemoglobin is conjugated with iron. Types and functions of proteins. Enzymes are biological catalysts used to speed up chemical reactions, like the breakdown of amylose by the enzyme amylase. Enzymes are proteins in nature. Structural proteins form part of the organism's bodies, like silk in spiders, collagen and keratin in hairs and nails, and muscles for humans. Signaling proteins are used to carry messages around the body. These are basically hormones. Although most hormones are made from steroids, a few are proteins, like insulin and glucagon that control blood sugar and growth hormone. Contractile proteins are involved in movement, like actin and myosin in muscles that enable it to contract. Storage proteins are biological reserves of ions and amino acids, like the albumin is a storage protein that is in the white part of the egg. Defense proteins are used to fight infections, like the antibodies. Transport proteins are used for transportation, like hemoglobin that transport oxygen and carbon dioxide. Proteins are also used as a source of energy, but proteins are only broken down to release energy in cases of extreme starvation. Now test for proteins. We have three methods of testing for the presence of proteins in a sample. The first and the most common one is the burette test. Here, we use copper-2 sulfate and sodium hydroxide. We will test for protein in the egg albumin. We take 1 ml of sodium hydroxide and add to the test sample. Then we add 2 to 3 drops of copper-2 sulfate to the sample and shake the mixture. You will see a violet color indicating the presence of proteins. Another test is called xanthoproteic test. Here, we will use concentrated nitric acid, ammonia solution, and we shall test for the same egg albumin. We also need a Bunsen burner. We take some 2 ml of the nitric acid and add to the egg albumin sample. Then we boil the sample in a water bath for 2 minutes. A yellow precipitate will be formed. Allow it to cool for some time. Take 2 ml of ammonia solution and add it to the now cool sample. The yellow precipitate changes to orange, indicating the presence of proteins. The last test is called the millions test. In this test, we will use a millions reagent and test for the presence of proteins in the same egg albumin. We take 2 milliliters of Million's reagent, add to the test tube containing the sample, and wait for some time. A pink color appears, indicating the presence of proteins. So we are done with the biomolecules. We look at enzymes. Now, most chemical reactions in the cells do not occur on their own or spontaneously. Instead, cells rely on enzymes to kickstart the chemical processes and proceed at a faster rate. Enzymes are biological catalysts that are protein in nature. A catalyst is a substance that speeds up or slows down a reaction without being itself changed at the end of the reaction. Ideally, enzymes, like all catalysts, work by bringing the reacting substances called substrates together, allowing them to react faster. Enzymes have a unique way of kickstarting reactions. They work by binding one or two molecules called reactants or substrates. Binding occurs at a special region in the enzyme called the active site. Once the substrate binds to the active site, they form what we call an enzyme-substrate complex. As the enzyme and the substrate begin to react, some of the chemical bonds in the substrates begin to weaken, causing them to link together. Eventually, a new substance is formed at the active site. This is the product. Once the reaction has occurred, the product is released from the active site. The enzyme returns to its original state and is ready to react again with another set of substrates. In chemical sense, all reactions require energy to kickstart them and drive them. Let's look at two reaction pathways, one without an enzyme and another with an enzyme. This is a pathway without an enzyme. As you can see in this graph, the reaction forms a product by following this reaction pathway here. This hilly part is called the activation energy, or the minimum amount of energy needed to start a reaction. Without a catalyst like an enzyme, the activation energy is quite large. Without an enzyme, an immense amount of energy is needed to drive chemical reactions in cells, which is very inefficient. Now let's see a reaction pathway with an enzyme. At first glance, you can already see that the pathway is different. With an enzyme, the activation energy is smaller. This means that less energy is needed to turn the reactants into product. If we combine the two pathways, we can visually see that the activation energy required for a reaction is lower when an enzyme is used. So basically, what enzymes do is that they lower the required energy to start and speed up the rate of a reaction. An example of an enzyme-catalyzed reaction is that in respiration. Don't worry if you haven't encountered it. You soon will. But usually, glucose reacts with oxygen to form carbon dioxide, water, and energy. The yellow curve represents the activation energy needed without a catalyst. The blue curve represents the activation energy needed with a catalyst. As you can see, the reaction is far more efficient when a catalyst is used. Properties of enzymes. Now, one major feature of enzymes is that they are very specific. These three enzymes clearly have different shapes in their active sites. So clearly, the kind of substrates they can bind are different. The thing is, every chemical reaction has an enzyme that catalyzes it. The specificity of an enzyme and substrate can be thought of as a key and a lock. The enzyme is the lock, and it will only react with the right substrate or key. Use the wrong key and the lock won't open. and the enzyme won't catalyze the reaction. This is called the lock and key model, and it's the best way to remember the specificity of enzymes to substrates. The second feature is that enzymes remain unchanged at the end of a chemical reaction. What that means is that they can be reused and that only a small number of enzymes is needed to catalyze a reaction. Enzymes are proteins. We said that in the definition. They are called functional proteins, meaning that they carry out specific functions. Next, enzyme cofactors and coenzymes. Now, cofactors are non-proteinous substances that activate enzymes. Most enzymes will not work without them. Some of the cofactors are metallic ions, such as those of iron, magnesium, zinc, and copper, while others are vitamins. Cofactors are required in small quantities, and like enzymes, cofactors are substances that can be reused again and again. Coenzymes are substances that affect the enzyme-controlled reactions. These are organic non-protein molecules that work in association with particular enzymes. Many cofactors are derived from vitamins. Now we look at enzyme inhibition. Inhibitors are substances that slow down the activity of an enzyme. There are two types of enzyme inhibitors: competitive and non-competitive. Let's see how each works. During a normal enzyme catalytic cycle, the substrate encounters an enzyme with specific active sites to which it binds, forming an enzyme-substrate complex. The enzyme then facilitates the breakdown of the substrate to its product, which breaks from the enzyme, leaving the active site free to catalyze another substrate as the cycle begins again. Competitive inhibition occurs when an enzyme encounters a blocker which mimics the characteristics of the substrate and binds to the active site. So when the substrate is encountered, the active site is unavailable for binding and no reaction will occur. Non-competitive enzyme inhibition involves the binding of a blocker on the enzyme but away from the active site. This binding, however, causes a conformational change in the enzyme, altering the shape of the active site, which prevents the substrate from binding, and so no reaction will occur. And now, factors affecting enzyme-controlled reactions. Now, before we go far, let's define something called optimal metabolic efficiency. This is when body cells are efficiently working, producing energy and removing toxins. Ideally, this only happens when enzymes' function is at its best. Factors that affect the enzyme activity include temperature, pH levels, and substrate concentration. Temperature. Every enzyme has an optimal temperature, and as you can see on this graph, the blue dot shows where the optimal temperature for this enzyme is. Optimal temperature means optimal enzyme activity until it gets colder or hotter. That's when everything starts working less efficiently. When the temperature is lower, the rate of enzyme activity is slowed down. And when the temperature is too high, the bonds holding the enzyme structure break. The active site loses shape and the enzyme falls apart. This explains the sudden drop-off on the right side of the graph. This is known as a denatured enzyme. Obviously, a denatured enzyme cannot catalyze the reaction it was meant to. Something similar happens when the temperature is too low, except that bonds don't break. The enzyme activity will occur but at a very slow pace. Simply said, too cold is bad, too hot is really bad. The other factor is substrate concentration. Remember, the substrate is the piece that is worked on by the enzyme. Substrate concentration works a bit different from temperature. Looking at this graph, you will notice that instead of the enzyme activity being represented by a dot, it is actually represented by a line. This means that once the substrate concentration reaches a certain point, the enzyme activity will stop increasing. The first point at which the optimal activity is reached is called the point of saturation. At this point, the enzyme activity is at its maximum and no amount of additional substrate will increase it. Let's say the point of saturation of an enzyme is 10 substrate units. Even if you increase that concentration to 100 or 1,000 units, the enzyme activity will remain the same because there is only a finite number of enzymes, and an enzyme can only catalyze a number of reactions one at a time. Holding all other conditions constant, the only way to increase the rate of reaction is to increase the enzyme concentration. Of course, less substrate concentration leads to less enzyme activity. Equally, low enzyme concentration slows down the rate of reaction. Lastly, pH levels. The pH graph is similar to that of temperature, and that is because it works the same way. Top of the graph means optimal pH level, which means optimal enzyme activity. If the pH drops, it becomes more acidic and the enzyme activity decreases. If pH increases, it becomes more basic and again the enzyme activity decreases. So overall, you will realize that it is important to maintain a constant internal body environment, particularly temperature and pH levels, in order to allow enzymes to work efficiently. But it is good to know that a constant internal environment leads to efficient enzyme activity, which results into optimal metabolic efficiency. This is why our body temperature is always at a range of 37 degrees Celsius. That temperature is a perfect balance for efficient enzyme activity. The last important substance to life is water. Now, life as we know it would not exist without water. Over 70% of the planet Earth is covered with water, and our bodies are 70% water. The structure of a water molecule and the fact that it is capable of forming hydrogen bonds with multiple other water molecules simultaneously gives water some very unique characteristics. These include the ability to act as a universal solvent, the properties of cohesion and adhesion, a high surface tension, a large heat capacity, and changes in density based on temperature. We look at each properties at a time. Water is often referred as a universal solvent, which means most substances dissolve in it. This has got to do with the polarity and hydrogen bonding ability of water molecules. When a salt such as sodium chloride is placed in water, the negative ends of the water molecules are attracted to the positively charged sodium ions, and the positive ends of the water molecules are attracted to the negatively charged chloride ions. This attraction causes the sodium and chloride ions to break up or dissociate in water. This is important since most of our cells is water. Those solutions of ions and molecules allow chemical reactions to occur much more efficiently and frequently. Water molecules are also cohesive and adhesive. The hydrogen bonds of water cause water molecules to stick together, a property known as cohesion. Polar bonds also give water molecules an adhesive property or the ability to cling to other polar surfaces. This is important because water fills the transport systems of plants and animals. Because of these cohesive and adhesive properties, water allows for efficient transport of nutrients and waste within an organism. Water also has very high levels of surface tension. Because water molecules at the surface are more strongly attracted to each other and to the air above, they cling tightly to each other. This surface tension explains why insects walk on water surfaces without sinking. Water has a very high heat capacity. This means that the many hydrogen bonds that link water molecules take a lot of energy to break for water to change its physical state, like from liquid to gas. This not only stabilizes the temperature of water bodies, it also plays an important role in the physiology of an organism. Since life is largely based on water, and the temperature of water rises and falls slowly, living organisms are better able to maintain their normal internal temperature and be protected from rapid temperature changes. Finally, the density of water is based on its temperature. Unlike many other compounds which contract when they freeze, the configuration of the hydrogen bonds of water causes it to expand when it freezes. This expansion causes ice to have a lower density than liquid water, and therefore ice floats on liquid water. If it were denser than liquid water, it would sink, making life below the water surface impossible. Instead, water bodies always freeze from top downwards. The ice at the surface acts as an insulator to protect water below from freezing. This allows aquatic life to survive during the cold winter seasons.